Hello everyone, my name is Eduardo. I am part of the Excellence in Breeding team, an arm of the Crops to End Hunger initiative. And this is a short tutorial that aims to introduce the audience to the use of the Breeding Scheme Designer tool, version 1. The outline of this tutorial is the following. We will review what is the scope of the tool, what are the major features of the tool, an example on how to estimate the predicted genetic gain, and also an example on how to compare different breeding strategies. So, the scope of this tool is to increase the familiarity of the breeders with the use and manipulation of the breeder's equation to estimate the predicted genetic gain and use it to compare different breeding strategies. That way, you can prioritize improvements that have a major impact on the rate of response to selection in your program. First, for a review on how breeder's equation is derived, please check the webinar on genetic gains available in the link projected at the bottom. But in summary, the breeder's equation defines how the distribution of a trait phenotype in a population of individuals that has a genetic basis can be modified by applying artificial selection. The change in phenotypic means between a selected population and the original population defines the so-called selection differential. Such change has to be multiplied by the heritability to change that to units with genetic basis. The change is also called the within generation change. When the selected individuals are mated and reproduce, the expected mean of the new population is the genetic mean of the individual selected. This change is called the intergenerational change. The breeder's equation has two popular parameterizations. One is defined in terms of the selection differential and the heritability. The other one is based on the accuracy, the square root of the heritability in the simplest case, and the selection intensity, or standardized selection differential, and the squared genetic variation. The Module 2 of Excellence in Breeding proposes the use of quantitative genetics principles as a mean to achieve greater rates of genetic gain. Some principles are not easy to accept by breeding teams until their short, medium, or long-term effects in the breeding program have been shown. Because of the cost involved, the team of quantitative geneticists from Module 2 worked mainly with simulations to provide recommendations on how to increase the genetic gains through proven methodologies, for example, new experimental designs, analytical methods, among other methods. Simulation can be done in two ways, through deterministic methods such as playing with the terms in Breeder's equation, but being limited most of the times to single generation and single trait analysis, and also stochastic methods, where much more flexibility can be incorporated and is not limited to single generation or single traits. Here we present a table of pros and cons. The Module 2 of Excellence in Breeding uses stochastic approaches to optimize breeding schemes, but these are made in a close collaboration with the breeding teams to incorporate all the complexities faced by the program, but are not the focus of this tutorial. The Breeding Scheme Designer tool is focused on the deterministic approaches, since these are quick to learn and can be used to predict genetic gain and compare simple strategies. Version 1 of the Breeding Scheme Design tool was designed in Excel by Gary Atlin, an officer from the Bill and Melinda Gates Foundation and experienced plant breeder. The version 2, available in the same web page, is a GUI developed in R Shiny with similar capabilities by Excellence in Breeding. Both tools use formulas based on published literature on quantitative genetics to estimate the rate of response to selection and can be used to compare the effect of different decisions in the breeding strategy. The Excel version is formed by 6 major sections. The input of time parameters that affect the denominator in the breeder's equation, and you can find them in gray color. The input of genetic variance parameters that affect sigma or the genetic variance that you can find in the Excel sheet in green. The input of crossing, evaluation, and selection decisions that affect the accuracy and the standardized selection differential or selection intensity in different levels of blue depending on the stage. the input of cost parameters that affect the final cost per unit of genetic gain and can be found in yellow in the Excel sheet. Intermediate calculations done by the Excel can be found in pink. And the final resulting parameters in white for genetic gain and genetic gain per dollar invested. The rows represent different scenarios or potential breeding strategies that you could compare. For the time being, let's focus on a single row. The columns that the user should focus on are the ones that require input. That is the gray ones, the green, the blue, and yellow. You can also find different tabs. The first one has the calculations for genetic gain. The second one has instructions on what exactly the column names mean. And the third one has a quick planner where you can actually plan different breeding strategies. Let's run some examples using the actual tool. Let's use the tool to calculate the rate of response to selection or genetic gain for a scenario where a breeding program has one season per year, it derives lines at F2, and it has 3 testing stages, which implies that for this crop, it takes 6 years to complete a cycle. You also have to provide the variance components. Here, they are expressed as ratios relative to the main genetic variance. Then in blue, you have to provide the numbers for the different evaluation and selection decisions. For example, how many locations, how many reps, how many individuals are actually being evaluated, and you can continue with the rest of the stages to indicate how many individuals are actually being selected. Depending on the number of stages that you are actually using, you may have to provide those numbers. Then, if you actually want to get the genetic gain per dollar invested, you will need to also provide the cost for the different decisions that you have specified. If you want to get more detail on what exactly means the different column names, you can always check the Instructions tab. Once you have provided all the numbers in the blue cells and the costs in the yellow cells, you don't have to worry about the pink cells. These are just calculated formulas that have been provided by the developers to calculate the final rate of genetic gain. You just have to move to the right and you will find the 3 final columns. Let's make a zoom. You can find first what is the total gain per cycle. This is expressed as the standard deviations of genetic gain. You also have the genetic gain per year, which is just the total gain divided by the number of years that takes to complete a cycle. And then, given that you have provided the costs, you can also get the cost per unit of genetic gain in standard deviations. Given the different input parameters that you can provide, you can always compare different breeding strategies. For example, you can go back and create a different row. Maybe what you want to compare is what is the expected impact if you actually derive lines 1 generation later or 2 generations later, etc., and so on. That, of course, will have an effect on the number of years per cycle, but also may have another impact on things like accuracy and intensity. You can play with the different variables, creating as many scenarios as you want, and you can just go to the end of the Excel sheet and look at what is the total gain. Of course, the more resources you invest, you will have more gain, but you may want to actually check what is the cost per unit of genetic gain. It may turn out that some of the smaller programs have more gain per dollar invested. Okay, that's all for this tutorial. Thanks for your interest in the Excellence in Breeding tools for optimizing breeding schemes. If you have questions about the tool presented, please reach any of the members from Module 2. We will be happy to support you to understand better how to use this tool in case this tutorial has not been enough. Take care and have a great day.